Okay. I will speak as clearly as I can, and if you do miss anything, type in the chat box and I can repeat myself. All right. So cells. Cells are the smallest living unit. There are things that are smaller than cells, but as soon as they get smaller than cells, then they're not considered life. So of course we have organelles that are smaller, we have atoms and things like that, but those aren't considered life by themselves. Most cells are microscopic. We do have some organisms, like this one guy right here, that the whole cell is an organism. And so some of those we can see with our eye. We can't see them with this much detail, but most of them we need a microscope even to see that they're there. Discovery of cells. Robert Hooke discovered cells, and he discovered them by looking at a sliver of cork, and cork is alive because it comes from a tree. And he saw these little empty-looking boxes, and he called it a cell because it reminded him of the cells that monks live in monasteries. And here we have a picture of skin tissue, and we can see the edges of the cell membrane, and then we can see the little dark dot in there, and what is that? Good. It is the nucleus. And then whenever we're under a pretty low magnification, this is pretty much all we can see. Once we get up much higher, then we can see some of the other structures. But that's normally all we'll see with a light microscope. So, principles of cell theory, we have 3 parts of the cell theory. The first is that all living things are made up of cells. The second is that the smallest living unit of structure and function of all organisms is the cell. We already talked about that. If once we get smaller than the cell, it's not considered life. And all cells arise from pre-existing cells. And before they discovered this, before they had microscopes and were able to see them, they thought that life just like spontaneously happened because it was so small that they couldn't see it. So then once they got more advanced technology and had microscopes, they could see the cells. And so now we know that all cells arise from pre-existing cells. They can't just poof into life. So this slide here is important concept in your project 2, the cell size. And cells have to have a large surface area to volume ratio. The surface area represents the cell membrane, and the cell membrane is how things come into the cell and leave the cell. And then the volume represents all of the stuff inside the cell, all what's inside the cell membrane. So here we have a picture that we just have on the left, one cube that's 2 centimeters by 2 centimeters by 2 centimeters, so that represents just one cell. And then on the right, they divided this same thing but into smaller pieces. So it actually is 8 separate pieces. So for the left, the total surface area, the area of all the outside parts, is 24 centimeters squared, and the volume is 8 centimeters cubed. So this gives it a 3-to-1 surface area to volume ratio. The one on the right, because it's divided up into smaller ones, we still have the same total volume, because you can see they still take up the same amount of space. But the surface area, because we've divided it into smaller pieces, is double. It's much larger, so it's 48 centimeters squared. So our surface area to volume ratio is 6-to-1. So this is going to be a lot more efficient at getting nutrients in. Nutrients have to go all the way across the whole cell. So when a cell is bigger, it isn't able to do that. So whenever our cells start getting big, they form into more cells and they split. If a cell gets too big, it can't survive. So characteristics of all cells, no matter what kind of cell it is, they all have these things in common. And we can see we have some probably cheek cells right there in that in that picture. So they all have a surrounding membrane. They all have cytoplasm, which is kind of the jelly that the other stuff is located in. They all have organelles, which are the structures for cell function, and they all have DNA. We have two main, if we were to split all of our cells into two main groups, we have prokaryotic, and karyo is the nucleus. Pro means before, so prokaryotic are before the nucleus. And these are going to be our bacteria cells. And then we have eukaryotic. Eu means true. So those are going to have a true nucleus. So we said all the things that all cells have in common. Now we're going to look at the differences between what the prokaryotic has versus the eukaryotic. So prokaryotic cells, these are before a nucleus, so they don't have a nucleus, and they also don't have membrane-bound organelles. So we'll see that the DNA is still in there, but it's just kind of condensed in the central region. We call that the nucleoid, but there's nothing actually containing it. And then the organelles are not bound by membranes, and we can see the only organelle that we even have is the ribosome. And then we have our plasma membrane, or our cell membrane, and our cell wall. And here we have a cartoon picture of bacteria, and then here's an actual under a microscope. You know, the ones that, because it has a flagella, it does look like a sperm. There are other types of cells that have flagellum that aren't sperm, but that's kind of the one that is the most common to us. But we can see in the actual microscope picture, we can see a flagella, and those are not sperm. Yeah. No, you're right. That's normally what we associate with the sperm. But the microscope picture is magnified 47,000 times, and those do have a flagella as well. But those look more like bacteria. All right, so that was for prokaryotic cells, which is going to be our bacteria. Then we get to eukaryotic cells, true nucleus, and they're going to have a nucleus bound by a membrane. Our prokaryotic cells are only bacteria. Our eukaryotic cells are everything else. So it's our funguses, our protists, our plants, and our animals. And the eukaryotic cells possess many organelles, whereas in the prokaryotic, all we had was just ribosomes. And this is a picture here of a protozoan, which is actually a single-celled organism. So the whole organism is only one cell. And I believe that's in one of your discussion questions is it wants you to look at comparing the differences between multicellular organisms and single-cellular organisms, or the advantages and disadvantages or something like that. So this would be an example of a single-cell organism. Anything that takes more than one cell to make the organism would be multicellular. So here we have a picture of an animal cell. We can see there's a lot more going on than in that prokaryotic cell. We have ribosomes, we have all of these different organelles that have membranes around them. And we're going to go through all of these different ones. So I know this is a lot just looking at it all together like this. Yeah, you don't have to draw that, Julie. This is made by some kind of a computer animation, so. And then our plant cell, also we can see there is a lot of different organelles, but we can see just looking at them, we can see a difference between our plant cell and our animal cell. Our animal cell usually don't have a certain shape, but our plant cells always are like rectangular. And that's because of the cell wall. They look completely different, yeah. They have a lot of the same stuff in them though. But if you're just looking at a microscope picture, you can always tell the difference because the plant cell has that cell wall. So they're always going to have a very rigid structure. Okay, so now we're going to go through, or does anybody else have questions before we move on? Chloroplasts, correct, are only found in plant cells. Okay, so we're going to go through our different organelles. First, we have our plasma membrane, which is another word for the cell membrane. Those can be used interchangeably. It's always going to be our layer on our plant cells. It's directly inside the cell wall. On our animal cells, it's the very outermost layer, and it's a double layer of phospholipids and proteins. And next unit, we'll actually spend several lessons just looking at the plasma membrane, because there's a lot of complicated processes that go on there. So how things move across the plasma membrane, like I said, we'll get more into this next unit, but some things are able to move freely. They can just go in and out, and those include water, carbon dioxide, ammonia, and oxygen. And then other things are not able just to move freely through it. They need help. So I like to think of it like a chain link fence. If you have something small, like a marble, you could just throw it straight through the chain link fence. But if you had something big, like a book, you wouldn't be able to just put it through. You'd have to either find something to transport it through, or you'd have to find like a gate or a hole in the fence or something. So some things can go straight through, other things have to be like escorted or have to find a certain channel to go through. For your fence, you could throw it over. Okay, so this is just a quick view of that. You don't have to know all of this right now. We'll do it next unit. But just the basic of what the cell membrane does is it lets some things pass through and it keeps other things out. So it's called semi-permeable. So like small uncharged molecules can go straight through. Lipid-soluble substances can go straight through. But then water-soluble substances or charged substances like ions can't. They have to be regulated. So our membrane proteins, we have channel and carrier proteins, which some of those molecules that are too big or that are charged, it can help them so that they can make it through the cell membrane. We have receptor proteins, which have a little like antenna looking guy on them, and they are looking out for just a specific chemical. We have cell recognition proteins, which identify a cell type. And then we have enzymes, which catalyze production of substances. And a enzyme, sorry guys, what was the second on? Back here? Okay. So, and like I said, the whole next unit, we have a whole chunk just on the cell membrane. So if this doesn't all make perfect sense for the cell membrane part right now, don't worry, just know what the purpose of it is. And then next unit, we'll go through really all of the processes and more specifically what the little parts of it do. So it does talk about enzymes though, and what enzymes do is they speed up a reaction, or sometimes they just make it so the reaction can get started. So here we have enzymes. So in this, our enzyme is our blue part, and the solid line here represents, this is how much energy a regular reaction without the enzyme, how much energy it would need so that it could get started to kind of get over that hump so that it could make the product. What the enzyme does is it lowers that amount of energy. So this reactant forming together to make this product, they have to have a lot less energy to go into it to get the reaction started, which means it's going to happen faster. So they lower the activation energy. For an uncatalyzed reaction, this is the activation energy. with the enzyme, this is the activation energy. So this has to put less energy in to get started. And then after this product is made, the enzyme will just attach and it will go catalyze another reaction. So they're not used up. Does anybody have questions on that part? Okay. Any time you do have questions, make sure that you ask them, okay? Or you can wait till the end if you feel more comfortable with that. Okay, so cell walls are found only in plants, in fungi, and in protists, and then we also see them in bacteria, but now we're just focusing on our eukaryotes. It goes outside of the plasma membrane, and in plants, this is what gives it that very rigid, specific structure that they always look. Oh, protists it's a kingdom of organisms. They're like amoebas and paramecium, some different algae. So we have all of our kingdoms. We have bacteria, we have animals, we have plants, we have protists, and we have fungus. So those are the ones that most of them are single cell. And they're microscopic. Usually a lot of them are in like water. So the cell wall is made of cellulose, which is what makes it crunchy. If you've ever snapped a plant, why it's crunchy is because of the cellulose. The reason they have that cell wall is because plants take in lots of water. And if it didn't have that cell wall, that strong thing holding it in, what do you think might happen to the cell? If it took in all that water and it didn't have a cell wall. It would leak out, okay? Actually, it can pop. The cell can burst. It would break, exactly. So they have to have the cell wall because they have this really big vacuole in it so that they can take in water. And that's the reason animal cells don't need that. Animal cells need to be more flexible. And we don't have a big vacuole the way a plant cell does. Next, the cytoplasm. It's a viscous, which means it is kind of thick. Like viscous meat is a substance's resistance to flow. So if you think of like honey is very viscous because if you dip over the honey jar, it takes a long time for it to come out. So it's a viscous fluid containing organelles. And what the cytoplasm is made of is filaments and fibers, a fluid called cytosol, and then all of the organelles are in the cytoplasm. Viscosity, yes, it's the resistance to flow. Animal cells do not have a cell wall. Humans are animals, so humans do not have cell walls. And actually, so the cell wall, we said, well, in a plant, the cell wall is made of cellulose, which is why that's actually before, or I guess animals that have an appendix, that's what their appendix does, is it breaks down cellulose. So if you look at like an animal, like a pig, it has an appendix that works because it can break down cellulose. Our body, we can't break down cellulose, which is our appendix doesn't do anything. Okay, cytoskeleton, filaments and fibers. We have 3 different fibers that make up the cytoskeleton. We have microfilaments, microtubules, and then intermediate filaments. Microfilaments are going to be really thin and like strand-like, so I think of it as like threads. Microtubules are tubes, so they're little hollow tubes. And then intermediate filaments are like kind of an in-betweeny. And the function of the cytoskeleton is they offer support, they anchor the organelles, they help substances move through the cell, and then also when we get into cell division, we'll look at these little structures called centrioles, and centrioles are made up of 9 microtubules. So here's a picture of, we can see actin, which is the subunit of microfilaments. We can see that here. It doesn't really look like any structure. And then our intermediate filaments, we can see they're a little bit thicker, so they look more like something. And then our microtubules, they're a lot thicker, and they're like little tubes. Cilia and flagella provide motility. Cilia are short and are used to move substances outside human cells. And flagella are whip-like extensions, and we talked earlier, that's what we find on sperm cells. So we can see our cilia, little hairs, and they're usually all around it to help it move, and a flagella is like a whip. And obviously this organism here has more than one flagella. Okay, so looking at our organelles that have membranes, so these are not going to be in anything else except for eukaryotic cells. We have our nucleus, which is the control center. It has a double membrane, and it contains chromosomes, which are the unit of heredity. When the DNA winds up super tight, the structure is called a chromosome. And then it also has the nucleolus. The nuclear membrane surrounds the nucleus, so it's what separates the nucleus from the rest of the cell. And it has pores in it because RNA has to pass out of the nucleus to go to the ribosomes to make proteins. We'll talk about that in a later unit. But those pores are so that the RNA can leave. But DNA is trapped in the nucleus. He's like a prisoner. DNA is heredity material, and chromosomes, we said, are DNA proteins, and they form for cell division, and proteins is spelled wrong, sorry. So when DNA is unwound and is just in like the long stringy form, it's called chromatin, and then whenever it coils up super tight to make what those little X's that we always think that chromosomes look like, then they're called chromosomes. The nucleolus direct synthesis of RNA, and the nucleolus is responsible for making ribosomes, and then ribosomes make proteins. RNA is another nucleic acid. We have DNA and RNA, and it is single-stranded. How DNA is always double, and that's what it makes that little helix. RNA is single-stranded, and RNA has a different sugar and a different base in it, but RNA is what takes the code from the DNA out of the nucleus to the other parts of the body. And we'll learn more specifics about that in, I think it's Unit 5. So you don't need to know too much about it now, just it's a nucleic acid. That's what the Na stands for, is nucleic acid. And it's the nucleic acid that can leave the nucleus. The endoplasmic reticulum helps move substances within cells, and it's a network of interconnected membranes. So this is like the highway system of the cell. We have two types, the rough ER and the smooth ER. The rough, the reason it looks rough is because it has ribosomes attached to it. And the ribosomes manufacture proteins. And we also have ribosomes that are not attached to the rough ER. We have some that are just floating in the cytoplasm. And the endoplasmic reticulum is always like right on the, next to the nucleus. So it kind of helps us to identify it. But it's like the highway system. It's how things get transported around. And then we have the smooth ER, which does not have ribosomes. And it has enzymes that help build molecules like carbohydrates and lipids. So it'll look the same as the rough ER, but without the little dots on it, which are the ribosomes. Okay, next is the Golgi apparatus. It is involved in the synthesis of the plant cell wall, and it packages and ships things throughout the cell. So it's like the UPS that the endoplasmic reticulum will move things out, and then the Golgi apparatus, they'll go through the Golgi. Actually, I think that's on the next slide. No, sorry, it's on one of them. So Golgi apparatus function, molecules come in vesicles. The vesicles fuse with the Golgi membrane. The molecules may be modified by the Golgi. It is a lot of information, Emily, and this class goes very fast. So that's the reason that, because it's online, you can use your notes for your tests and stuff. So always just take good notes, but it is a lot to remember. Then the vesicles leave the Golgi, and the vesicles can combine with the plasma membrane to secrete contents. And there's a picture on the next one that the pinched off part is, we call a vesicle. So we have the rough ER, it has these ribosomes on it, and the ribosomes make proteins. Then it exports the proteins, and they go to the Golgi. Then our proteins move through the Golgi, and as they go through the Golgi, they're modified and sorted. And then they pinch off from the Golgi, called a secretory vesicle, and they get moved to the cell membrane. And then once they get to the cell membrane, they fuse with the cell membrane, and then it kind of pushes it out. Okay, so the Golgi is like UPS. It packages, it sorts, it modifies, and then it ships it out of the cell. Not the DNA out. The DNA is stuck in the nucleus. It can't leave. It's to let the endoplasmic reticulum make proteins, and then it sorts and modifies the proteins, and then it ships that out. And it can do it with other substances as well, carbohydrates and lipids, but not the DNA. The DNA is the one that it's stuck in the nucleus. It's a prisoner there. Lysosome they contain digestive enzymes, and they are kind of like the little digestion centers. So think of it, it's like the stomach. So they aid in cell renewal, stuff that needs to be like old cell parts. It digests them, and then once they're digested, then they can be shipped out of the cell. But if you lyse something, that means that you break it open. So lysosomes, they break down stuff because they have digestive enzymes. I'm not understanding your question. It can do all these things for the plant or whatever. Uh, this here is an animal cell. It's a eukaryotic cell. And there's a lot of other stuff in the cell. This is just showing the parts that are for this process where the Golgi is packaging and shipping stuff out. So this is not creating nutrients. Animal cells can't create nutrients. They have to get energy from consuming other things, and that's a different process. It's in the mitochondria, so we haven't gotten to that one yet. And then in plants, it'll happen in the chloroplast. So this isn't creating nutrients. This is kind of creating parts to make more cells. Because our cells age, and so we need to make, or they get too big, so we need to make new cells to replace old cells. Yeah, so we think of proteins as like meats, but these are proteins much, much smaller scale. This is to make new cell parts. This isn't to get energy. This isn't like us consuming proteins. So this would be making the proteins in other organisms, and then we eat those other organisms. But that process takes place somewhere else. Okay, so for this slide, all we need to, okay, you can, if you can think of more what your question is later, Christopher, we can come back to that. So for this one, we just see that the rough ER has ribosomes, the smooth ER does not. They have these ribosomes on them. The ribosomes are made in the nucleolus. Ribosomes make proteins. They travel the rough ER, it's like the highway system. Then they go to the Golgi. They go through the Golgi and are packaged and shipped out because the Golgi is like the postal system. Lysosomes, we said, digest things. So if we have cell parts that are old that they need to be replaced by new cell parts, the lysosome will break them down so that they can be exported out of the cell. Vacuoles contain water and nutrients and waste for the cell, not for it's not part of digestion. It's easy to get it confused on a microscopic level with the macroscopic level. So the lysosome is digestion like within the cell. It's not like our digestive system. It's to get rid of old cell parts. Vacuoles store these things. We see more of them in animals. There's usually multiple ones in animals, but they're very small. In plants, we normally have one giant vacuole because plants store water. And this is the reason, so we can see on this one here, this whole big giant thing here, this is the vacuole. And so we have to have a cell wall because if that vacuole gets too big, if we didn't have that cell wall to have it keep its shape, it would burst and the cell would die. Yep, they will. You cannot survive without it. Some organelles are bacteria-like in kind of the way that they, let me see how to explain this. You know what? Don't worry about that. Not important. We'll just go through more types of organelles. We have the mitochondria and the chloroplasts, and these are sort of function in, is that an eyeball? No, it's not an eyeball, it's nucleus. In energy, so mitochondria release energy, and we see lots of those in animal cells. Animals cannot make their own energy. We have to get energy from things that we consume. And in a chloroplast, we'll see those only in plant cells because plants can make their own energy. So both cells will have mitochondria. Only plant cells will have chloroplasts. And the chloro means green, so they're easy to identify because they're green. The mitochondria have their own DNA. It's called mitochondrial DNA. And they're bound by a double membrane. Here's a picture of one. It has these kind of like folds in it to increase the surface area so that more energy production can happen there. So think the mighty mitochondria, so that's what makes the energy. It's like a Red Bull for your cells. It breaks down fuel molecules in cellular respiration, and it releases energy as ATP. And this energy is what fuels our other cell processes. Chloroplasts are derived from photosynthetic bacteria, and they capture solar energy. And like I said, they look green. It kind of has these little stacks of pancakes in them, which is called a granum. And we can see in the actual cell picture, we can see that granum. And it has a double membrane. We'll only see them in things that are photosynthetic. So there are some more things that are photosynthetic than plants. We do have photosynthetic bacteria. Photosynthesis is the process that takes place in the chloroplast, which makes cellular food. It makes glucose using carbon dioxide and water and sunlight. And we'll look a lot more at the cell respiration and the photosynthesis in the next unit. Glucose is sugar, it's a simple sugar. So, have a table that compares all of them. Cell wall can be in bacteria and plants, but not in animals. All of them have a plasma membrane. Any of them can have flagella or cilia, depending on the specific type of cell. Endoplasmic reticulum is only in plant and animal. Microtubules are only in plant and animal. Centrioles are only in animal, and we'll talk more about those. I don't think they even have them in this unit. The centrioles we'll talk about when we get into cell division. The Golgi is in plant and animal. Nucleus is in plant and animal. Mitochondria, plant and animal. Chloroplasts, only in plants. Chromosomes, in bacteria, we just have a circle of DNA. In plants and animals, we have chromosomes, and they're located in the nucleus. Ribosomes are in all of them. Lysosomes are only in our eukaryotic plant and animal. And then vacuoles will have usually small ones, but there's more than one in animal, and then we usually have just one giant one in plants. Okay, so I know it's a lot of information. That's why those what should I know things, that's the reason we do those. There is in the